we would be at this every day, and I very much am. Yesterday, we did this in 20 minutes. This is the entire topic of osmoregulation. So we covered ultrafiltration, selective reabsorption, osmoregulation, and then we also covered the effect of ADH on osmoregulation. My ears are doing this thing where it's like blocking out all of the ambient noise, which is actually very helpful so I can focus on my voice. I don't know how long we're going to be live for today. I'm hoping not too long, because what I'm noticing now is the more we're doing these streams, the less I'm streaming for, because we can jam through an entire topic in 20, 30, 40 minutes when it would usually take us hours and hours. So there are a handful of topics that I really do want to focus on. One of them is gas exchange in insects and fish, primarily because that is something I have not revised probably since AS levels. I want to cover in general meiosis, because that is honestly the worst topic ever. I hate that so much. I also want to cover the topic we're still doing now, which is DNA. Specifically, things like transcription factors. I know what they are, but I don't know how to explain them. So, let's crack on with gas exchange. We could probably talk about this actually very quickly, just to make sure I haven't wasted revision. Ultrafiltration. So this is the kidney nephron. The Bowman's capsule, that's the I don't know how you pronounce it, glomerulus, I don't know, leads into the proximal convoluted tubule, which is then leads into the loop of Henle, the distal convoluted tubule, and then the collection duct. In this general area here is where ultrafiltration occurs. We have the afferent arteriole, which comes into the capsule itself. This is at a high pressure, which means it's going to force all of the waste substances out of the arteriole, out of the capillaries, and into the capsule. Selective reabsorption occurs here of a proximal convoluted tubule. What you have is it's basically co-transport just on crack. You have to reword a couple of things, but it's literally co-transport. Ensuring that glucose and things like amino acids are put out. Finally, in the loop of Henle, we have osmoregulation. This is very easy. Ions out, water out, ions out, water out. Basically, the ions are actively transported out. Things like sodium ions, chloride ions. That then raises the water potential, therefore water leaves. On this side, the permeable side. Because there's less water, the solution is far more concentrated with ions, so ions are going to move out, firstly via facilitated diffusion, but then by active transport, which once again raises the water potential, forcing water out, and thus a lot of water is lost, a lot of ions are lost to the interstitial fluid, which then goes into the capillaries, where things like the water are recycled. Meanwhile, everything else is going to travel along into the collection duct. In the collection duct, the effect of ADH is that it's secreted in the pituitary gland, it's detected by the hypothalamus, and the way this works is it binds to receptors on the collection duct cells. It activates an enzyme called active phosphorylase. This enzyme causes aquaporins inside the collection duct cell to fuse with the cell surface membrane, increasing the permeability of these collection duct cells to water, increasing the amount of water that is lost in the collection duct and is re-entered into the bloodstream. That is, we did maybe a 20-minute stream on that, and I've just summarized it all in like 3 minutes. So, there you go. So, gas exchange is something I actually have rarely revised, to be honest. Gas exchange in humans is something I feel somewhat confident on, mainly because I revised that a lot, but I don't remember much about the other two. So, gas exchange in humans, we'll do inhalation first. Oh my god, why don't I remember any of this now? Also, well, I keep losing my pen. So with inhalation... with inhalation, the whole idea with that is that it increases the volume in the thorax, so therefore air enters down the... Is it a pressure gradient, I'm assuming? From the atmosphere into there, from an area of... Yeah, because if you increase the volume, you're lowering the pressure. So it's a pressure gradient. Okay, cool. I'm not insane. So how does the pressure get increased? Well, decreased. I don't actually recall. I know there's something about the diaphragm. Is it the diaphragm that's pushed? Does the diaphragm flatten? I also don't know how to spell it, so whatever. The diaphragm flattens. The rib cage is pushed up and out, I believe. I guess pulled. Rib cage is pulled up and outwards. The effect of both of these is that it's going to increase the volume in the thorax, which overall is going to establish a pressure gradient because there is now a lower pressure in the thorax. Meanwhile, with exhalation, I would assume it would be the opposite. Like the diaphragm relaxes. I know there's something about it relaxing and then contracting slightly. Is it the diaphragm contracts slightly and then the rib cage is pulled inwards, which would increase the pressure, forcing air out? Let's have a look, shall we? Yeah, we might literally only be live for like 10 more minutes, because if gas exchange in insects is as easy as I'm remembering it to be, we might be fine. Diaphragm muscles contract, flattening the okay, it's... Oh, it's a lot more than I'm remembering, actually. Okay. The diaphragm muscles contract, flattening the diaphragm. So I missed a tiny bit there about the diaphragm. The muscles contract. Then, the bit that I forgot is the... what do you call them? The intercostal muscles. It's the external intercostal muscles, which are gonna, I'm assuming, contract. Okay. So the external intercostal muscles are gonna contract. This will pull the ribcage upwards and outwards. The diaphragm muscles contract, causing the diaphragm to flatten. Okay, so I missed two details. In terms of exhalation, it's the diaphragm muscles contract. So no, relax, sorry. Don't mind me, I'm a bit thick. External intercostal muscles relax. Decreasing volume. So it is literally... Okay, cool. It is literally just relaxing. So in inhalation, the external intercostal muscles contract, leading to the ribcage being pulled out. The diaphragm muscles contract, leading to the diaphragm flattening. In exhalation, it's the opposite. The diaphragm relaxes, the external intercostal muscles relax, which will decrease the volume in the thorax, which will increase the pressure gradient, so air moves out. Cool, okay. So that's gas exchange in humans. Now this is the bit that I actually don't recall a single thing on. Gas exchange in fish and insects. I'm trying to recall a single thing about this. Let's see. We'll start with insects because I feel like that's the easier one. I know that there are the tiny pores on the exoskeleton. Those are the spiracles, right? Those are the spiracles. What are the other things? Tracheae and tracheoles, that's it. I'm just kind of drawing what I assume it to be. I also don't know how to spell this, bear in mind, I'm just kind of improvising. So spiracles, which are the, there's a word for them. I guess the pores on the exoskeleton. The pores on the exoskeleton. The tracheae, I wouldn't even know how to describe them really. It's just kind of the air moves from the spiracle to the tracheae, and then it branches off into the tracheoles. The thing I remember about the tracheoles is that each one is connected to its own cell. I remember that. Because there was an exam question however many years ago, where it might have been 4 or 6 marks for describing the properties of gas exchange in insects, and you would have literally gotten a mark for saying that. You would have gotten a mark for mentioning the tracheoles, and then branching off so that each tracheole reaches a cell. That's obviously important so that air reaches every cell to provide the air for respiration, and obviously then the CO2 to be released. What are some other properties? This is 1 cell thick. There is moisture at the tips of the tracheoles. I don't know whether it's moisture or water, I'll just put moisture. What that does, from what I can recall, is the water will move into the cell during possibly aerobic respiration. And I think that forces out air, or it forces in air. I don't recall, to be completely honest, because there's three ways that gas exchange happens in insects. The first is like passive, simple diffusion. The second one is where they rapidly contract and relax. And I'm guessing due to the pressure gradient that's established there, it forces in air very quickly. So I'll just put contracting. Or muscle contraction. And then the third one, I don't recall the name of at all. I don't recall how it works. So should I go and pull up a mark scheme and see if we can infer some information from there? It's topic 3, exchange of substances. Mass transport in animals. There's definitely 1 or 2 exam questions on this, I'm not going crazy. I'm not crazy, please believe me. Is it this? Okay, here. Wow, I was literally on the question last time. Tracheoles have thin walls, right? That was going to be... Well, I did put 1 cell thick, but... They have thin walls, so a short diffusion pathway. Obviously, that's the whole gimmick here, right? when you're talking about mass transport or gas exchange, you need to mention the word diffusion pathway, like your life depends on it. Highly branched, so short diffusion pathway. I mean, I guess I kind of spoke about that briefly, but obviously the fact that they're branched, short pathway. Because yeah, if it was literally a single line, then how is it going to reach cells over here? You know, it needs to be highly branched. I get what they're implying. Highly branched, large number of tracheoles, so large surface area. Again, pretty obvious. The tracheae provide the tubes full of air, so fast diffusion. Fluid in the end of tracheoles that moves out during exercise, so faster diffusion through air. Okay. So it moves out, into tissues. Okay. I guess we can infer that then. There's moisture at the ends of the tracheoles, and it moves into tissues. And all it explains there is that it increases the diffusion rate through the air to the surface, the gas exchange surface. Body can be moved by muscles to move air. So that mark scheme refuses to even mention spiracles, but one thing I remember about them is that they're able to open and close. So I remember there being an exam question where it was asking you about if there's more CO2 in the insect, what's going to happen? Obviously, you would say that there will be more spiracles open so the CO2 can diffuse out. Honestly, then, that's not bad. That is not bad at all. As long as you literally remember the idea, I guess we can write all the properties here quickly to remember them. The idea that the spiracles are highly branched, which increases the surface area / shorter diffusion pathway. They are this. Well, what does it say specifically? They have thin walls. Okay. I was going to say one cell thick, but I guess thin walls works. Which obviously is part of the short diffusion pathway. The moisture, which as we've just established, somehow the moisture moving from the tracheoles into the cells allows there to be an increased rate of diffusion. Also, just talking about the tracheae, which provides the tubes with air in the first place. So to be honest, that isn't bad at all. I don't think that is a particularly bad topic. I think literally the only thing that would catch me off guard is having to talk about those properties, because I just very quickly glossed over the idea of diffusion there. When this is the gas exchange topic, you can't just gloss over diffusion. Like, there's some topics in biology where you just have to glaze certain words so much, like, you know, talking about enzymes and the idea of ES complexes, complementary structure, tertiary structure. Like, it gets on your nerves how much you have to write the same things, and then the one time you forget to write it is the time you drop marks. Right, fish. We have I don't even know how you would draw this, to be honest. I'm going to draw it how I imagine it to look. You have, like, the gill filaments. The gill lamellae. The idea is that the lamellae, there's a lot. So, many lamellae increases the surface area. The idea of a counter current flow. That is something very important. The idea that blood and oxygen flow the other way. Water and blood flow in opposite directions. Water and blood flow in opposite directions. So blood is always passing water with a higher oxygen concentration. So in other words, a constant diffusion gradient is met. And yeah, if water and blood flowed in the same direction, the equilibrium would be reached. So it means highly oxygenated water is always passing blood. And they would allow you to draw a diagram, which is something I would do. Talking about 1 cell thick in humans, yep. Plants. I forget gas exchange in plants. We might have to say that for another time, because I would love to do a big in-depth talk about plants. Things like guard cells opening and closing, transpiration, translocation, things like that. Gill filaments and filamellae. Yeah, water and blood flow in opposite directions, which maintains a constant diffusion gradient along. Oh, you should mention along entire. What would you say? Along the entire length of lamellae. So there's your 3 mark points. If you want to get 3 marks very easily, mark point 1, mark point 2, mark point 3. Water and blood flow in opposite directions, maintaining a constant diffusion gradient across the entire lamellae. That is easy. A reduced surface area would obviously increase the distance of diffusion. You can get 3 marks just for talking about inhalation. There are about 5 or 6 questions. 5 or 6 questions purely on the countercurrent flow. So definitely drill that into your head. There is a question here. What is this question? Describe one feature of the alveolar epithelium that makes the epithelial cell well adapted. Do not refer to surface area and moisture. Easy. 1 cell thick reduces the diffusion pathway. Yeah, 1 cell thick reduces diffusion pathway. Easy. 2 marks. Well, there you go, guys. That's all the time that I really want to pour today. I've spent the last hour doing an essay, so I'm happy, to be honest, considering that only took 25 minutes. Yesterday's stream only took about 20 minutes. I'm happy to maybe do more than one of these a day if we can each time drill into a topic, because I find these revision streams fun. I can't even lie. Yes, God forbid I find revision fun. I find these streams fun. So what I might do is I'm going to obviously document this information on my revision cards. That's what I'm doing, by the way. After yesterday's stream, I made this card here, which is just talking about osmoregulation. Very crude summary of it. But I underlined the key mark scheme words, things like that. That's my goal now, is to make one of these after every stream. I could make 2 today. I could make 1 on gas exchange in fish, 1 gas exchange in insects. So yeah, cool.